All right. Thank you so much, everyone, for joining. Looks we had a poll on the top of the screen, so just go ahead and answer while we wait about what you're currently using to track your samples, and we'll finish it at the end of this slide. Just helps me understand a little bit more about where you're coming from, what pain points you might be using. But let's go ahead and get started. So thank you everyone for joining us today. I'm Lance, a product specialist here at LabKey. I help labs organize their samples and scientific data more effectively. Before this, I worked in organ and tissue donation, where we process thousands of biological samples every single day. And I saw firsthand how chaotic freezers can get when we have that many samples. So today we're going to be talking about something nearly every lab struggles with, but doesn't really talk about enough, and that's knowing exactly where your samples are and keeping that system consistently organized. Whether you're a scientist trying to quickly locate a critical sample for an experiment, or a lab manager focusing on maximizing freezer space and staying compliant, your freezer setup plays a huge role in your daily success or stress. So let's start with a scenario you've likely experienced. You need that one important sample. You think you have the sample ID in your spreadsheet, but the entry has a typo or two, so you're crossing your fingers and hoping it's right. You open the freezer and start digging. Minutes pass. Your gloves are freezing, your fingers hurt, the boxes are wet and slippery with condensation. You're pulling boxes out and putting them back. You're starting to second guess everything. Then comes the real danger. Your samples are beginning to thaw. You accidentally knock over a rack, maybe a whole box spills. Your experiment is delayed or derailed simply because you can't find what you need. And every extra minute you spend rummaging puts the rest of your samples at risk. In the worst case, you break your freezer. Or you realize a freezer failed overnight. You come back the next morning to see a puddle underneath your freezer and you've lost thousands of samples. This kind of chaos isn't just frustrating, it's expensive and dangerous to your research, but it's also avoidable. So let's go ahead and kind of look into everything and what we can do and the kind of the benefits of organizing our freezers. And let's look at our poll really fast. So it looks like a lot of us are using spreadsheets, freezer management software, and it looks like some pen and paper. So I'm glad everyone that's actually answered is at least using something to manage their samples, but we'll kind of go into greater detail and kind of the best practices for those, as well as some just physical tools you can use too. So thank you so much for those who participated. So let's talk about why organizing your freezer matters in the first place. You're probably here because you already feel the pain, whether it's lost samples, wasted time, or frustration across your team. But let's break it down. Time is money. Every minute a technician spends digging through freezers is time you're paying for, time they could be spending on experiments, analysis, or high impact work. Uh, Good freezer organization helps to reduce that time that you're spending on things that shouldn't be really spent time on. It also helps to reduce costly errors. When your freezers are disorganized, it's easy to grab the wrong sample or not find it at all. Good organization gives you confidence in what you're working with. You'll also know what can be discarded. When samples are expired or are no longer needed or fall below volume thresholds, they should be cleared out. Good organization helps you identify these and free up critical space. It also protects your equipment. Less rummaging means less time with freezer doors open, and it reduces frost buildup and wear. This means fewer breakdowns and fewer costly repairs. Ultimately, good freezer practices don't just make your day easier, they support compliance, productivity, and the long-term sustainability of your lab. So if the benefits are clear, why haven't most labs already solved this? Let's walk through some of the most common issues I see in labs across research, clinical, and academic settings. First, there's disconnected records. Often, sample data lives in multiple spreadsheets or across multiple systems. There's one for locations, one for metadata, maybe more scattered across shared drives or individual computers. It's nearly impossible to get a clear picture without manually cross-referencing across all these different systems. There's also complex and inconsistent freezer layouts. Freezers can have multiple compartments, drawers, and boxes in different shapes and sizes. When you track locations in a freeform way, like writing box 2 without any context, mistakes happen. There's duplicate entries, misplaced samples, and confusion's very common. There's also untracked sample movement. Samples are constantly being taken out, moved to new boxes, or disposed of. But if movements aren't documented, immediately and accurately, you lose track of where the sample actually is or if it even still exists. There's also not a standard disposal process. Many labs hold on to samples that should have been discarded. They're either expired or empty tubes. These take up space. Without a clear policy and a system to enforce it, clutter builds, and it becomes harder to know what's usable and what's just taking up freezer space. There's also cumbersome search processes. When it's time to retrieve a sample, the process can be a nightmare. You're searching through multiple spreadsheets, you're dealing with typos or inconsistent naming, and you don't really know if the physical location is still accurate. Even if the data is there, if it's not clean and searchable, it might as well not be there. All of this kind of adds on to a compliance and audit risk. If you don't have an audit trail that's tracking movement, it's hard to carry out your investigations. If you can't track that sample movement, ownership, or disposal, it's also jeopardizing your lab's certifications, funding, and regulatory standing. All of these issues build on top of each other and can create major consequences. But the good news is that every one of these problems can be solved. You can centralize your records, so stop using 5 spreadsheets to track one freezer, move to a centralized system that combines sample metadata and storage locations into one place. You want to define your freezer hierarchy. So create a consistent structure, like you go from a freezer to a shelf to a rack to a box, and stick to it. This clarity helps to prevent duplicate locations and make searching easier. You also want to track sample movements in real time. You want to implement check-in, check-out processes for samples so every movement is logged, who moved it, when, where it went, and why. And you'll also want to use clear rules for when you should remove samples from storage altogether and set a clear and regular review process. Your software should be able to flag samples automatically based on that disposal criteria. You want to make your data filterable, so use structured column-based data. Avoid mixing information into single fields. Comments fields are notorious for this, and this makes it easier to query, search, and report on your samples. You also want to automate your audit trail. Every action from creation to movement to disposal should be recorded automatically. This process protects your team and simplifies compliance reporting. And these just aren't nice to haves. They're practical, achievable steps that will help transform how your lab manages samples. And it also eliminates the chaos we talked about earlier. But what are the tools to help kind of get to those solutions? So to improve freezer organization, we need to think in terms of 2 categories of tools. You have your physical tools and your digital tools. So let's start with the physical ones. You can use racks and shelving systems. Instead of just stacking boxes haphazardly, racks let you store boxes in fixed slots, either vertically or horizontally. Some examples can include cryo racks for standardized box sizes, and then there's also sliding drawer racks that pull out like a filing cabinet. There's no more searching in the abyss at the back of your freezer and hurting your back and knees because you have to dig around. Just pull it out much easier. You also want to try to use standardized boxes with dividers whenever possible. These are typically 9 by 9, 10 by 10, giving each tube its own defined slot. They come in sturdy cardboard or plastic. You just want to make sure that the lid stays on because they can be notorious for popping off and spilling all your samples everywhere, which is not fun. You can use rubber bands to help secure the tops of the lids. I would use 2. Don't use 1 because those can break in the freezers. 2 is always better. This is just a cheap and effective way to prevent those spills. You'll also want to implement labeling systems. Everything should be labeled. Samples, boxes, shelves, racks. Barcode labels are ideal, especially for samples, because they reduce data entry errors and are easier to scan. You'll want to use cryosafe labels or cover standard labels with clear tape to protect them from frost and condensation and keeps them from falling off. Ideally, you would apply labels before a sample is frozen. If a sample is already frozen, you'll just want a paper towel handy so you can wipe off any of that condensation. Also use color coding. Using colored tape or tags to differentiate between sample types or workflows can really help keep you organized. For example, red could be your discarding of samples. Blue is going to be for archiving samples. Green is for samples in active use. You can use this on the box or the rack that you're using, or even some highlighters on those specific samples. In my previous lab, we used these types of labels to quickly tell if a sample was going to be shipped back to a client, if we were going to be archiving them in-house, or if we were getting ready to dispose of them. Now, for digital tools, a lot of folks use spreadsheets. These are often the first step for many labs. They're familiar and inexpensive, and they work okay when your inventory is small. But as your sample count grows, spreadsheets become fragile and error prone. It's really easy to accidentally delete rows or overwrite data. It's difficult to collaborate on. They're prone to crashing as the file gets large, and there's no built-in protections for duplicate entries or formatting rules. Just this week, I was helping a lab clean up a spreadsheet with thousands of samples, crashed multiple times on me during the cleanup, which is a painful, time-consuming process that could have been avoided with better tools. So that brings us to more scalable digital solutions like LIMS or dedicated sample tracking software. So let's go deeper into what a LIMS is, or a laboratory information management system. These systems are designed to do much more than just store sample data. They provide end-to-end traceability and help you scale without chaos. So here's what a good LIMS system can offer. It offers sample registration and detailed metadata, so you can capture not just what a sample is, but everything about it, the donor information, the collection date, tube type, volume, and more. It helps you track the sample's location across an entire freezer structure from the freezer, shelf, rack, box, well inside that box. This gives complete visibility and eliminates guesswork. It can also help track your freeze thaw count. So many systems log how many times a system has or a sample has been thawed, which is key to assessing quality and preventing overuse. It also goes beyond storage. You can manage sample processing, shipping, chain of custody, and even testing workflows and tracking your assay data with those. It also has integrated ELNs or electronic lab notebooks. So you can ditch your paper records, you can capture notes and procedures, and link them directly with the samples involved. But I think what's most important with these limbs and what a lot of groups struggle with is having a comprehensive audit trail. A good limbs logs every single change, what was changed, who made the change, when it happened, and why it was done. This becomes incredibly important when samples go missing or questions arise during an audit. You can trace the sample's full history, for example, who last moved it, when they did it, and where they intended to place it. This level of tracking doesn't just support day-to-day lab work, it helps to ensure regulatory compliance, maintains data integrity, and builds trust in your results. Now, what actually makes a good freezer management software or a good LIMS, right? It has those features, but how do you know if it's actually going to be worth it? Because there are a lot of options out there, but not all of them are created equally. Your freezer map shouldn't be a standalone drawing or separate spreadsheet that you update manually. It should be directly connected to your sample metadata, who collected the sample, when, from whom, or what source, and what it's intended for. Some software solutions keep this information siloed. In those cases, you might see which samples are in a freezer, but not their history or data, or you might see a sample details, but you can't easily locate them in storage. I worked in one of those softwares. It was a nightmare trying to figure out where our samples were. But instead, you want to look for software that ties all your data together into one view. When you open a box map, you should immediately see exactly what each sample is, along with all of its relevant details. This context is critical for making confident decisions, reducing errors, and saving time, because more often than not, you'll see this kind of weird sample ID that just says blood 4733. That means nothing to me. I need these details to know what exactly I'm grabbing. And this image on this slide shows how at LabKey, we display this information right inside the box so you know exactly what you're grabbing before you even open the freezer. Next, let's talk about flexibility, which is often overlooked but extremely important. You should be able to capture every single sample type you need. You should be able to capture all of the metadata you need associated with your samples or with your donors or patients. But you should also be flexible with your freezers. Your software should be able to match your exact freezer configuration, no matter how complicated it is. You should be able to model as many different shelves, racks, drawers, boxes, or even bags as you actually use in your lab. Box sizes should also match what you have in reality, whether it's a 9 by 9, 10 by 10, 8 by 4, or something custom. Beyond that, labs are always evolving. You might add new shelves, reconfigure racks, or switch to a different type of boxes as needs change. Your software should be able to handle these adjustments without requiring a full rebuild or complex workarounds. Another key part of flexibility is permissions and roles. Not everyone needs the same level of access, right? Your system shouldn't let you your system should let you control who can update freezer layouts, who can move or discard of samples, and who can only view storage locations. This helps ensure your data integrity, prevents accidental changes, and supports a more secure and compliant environment. Finally, and I think what's most important, is the software has to be easy to use. If it's not easy, people aren't going to use it. Moving samples, discarding them, updating freezer layouts, these actions should be done in seconds, not hours. The user interface should feel intuitive, and even for like new users, right? They should just be able to pick it up very quickly. On top of that, strong customer support is critical. Ask yourself, does the software provider help train new users? Do they support you in configuring your lab setup? Are they available to answer questions when things come up? At LabKey, we provide our full customer support at no additional cost, which when we help get you started, we train new team members, we answer your questions at every step of the way, make sure that you're using the software and it's successful for you all. So that way you're not left to figure it out on your own. So once you have an idea of what tools you want to use, how do you go about actually implementing them and preparing your lab for them? Before you even start digitizing your freezer map, it's important to prepare your physical space first. You want to look at what you currently have and start understanding and clearly documenting your freezer layout. Outline the full hierarchy. You want to look at the freezer, the shelf, the rack, the box, the well, the sample. Having this clearly mapped out on paper or in a diagram will make your transition to digital tracking much smoother, but it also helps to point out where you might have some areas of improvement. You'll want to consider your physical organizational tools. Using racks, dividers, and standardized boxes can help reduce that chaos and make it easy to create clearly defined sections in your freezers. So you'll want to go back and add those wherever you see fit. This can help that instead of kind of digging through stacks of unlabeled boxes, you'll know exactly which shelf, which rack, and which box to go to without even having to read a label. You should still use them, but it makes it much easier to find things that you need. And don't forget about your bags too, right? These are for things that don't fit neatly inside those standardized boxes. You'll still want to document where they are. If you store samples in bags, make sure you plan space for them in your freezer map so they don't end up as unidentifiable mystery blobs at the bottom of your freezer. Once your data and your physical setup is ready, you'll want to start thinking about space optimization and maintenance. While mapping, you might realize you're holding on to samples you no longer need or won't need soon. So you'll want to create a discard policy to free up your valuable space. This could include looking for samples that are expired, samples from patients who have deconsented, or samples below a certain threshold volume. Decide on a discard schedule, whether it's weekly, monthly, or annually, and outline your approval process. Do you simply need a documented audit log of who discarded the samples, or do you require a manager level approval before disposal? Your software should make it easy to query for samples meeting these criteria and quickly generate a list of items to discard. Here, I just searched for samples that were expiring either today or before today and was able to pull up a list of everything that was still in inventory. So look at your software and see if it can do that for you. Next, you'll want to think about your freezer maintenance. A frosted over, messy freezer can make it nearly impossible to verify labels or maintain accurate maps. Regular defrosting and maintenance not only extends your freezer's life, but also keeps your data accurate. Make sure your software can support bulk moves of samples to other freezers or temporary storage during defrosting so you can maintain order even during maintenance. After you've tackled your physical setup, it's time to actually look at your existing data. Many labs are still currently tracking freezer inventory in spreadsheets, but these files can be messy, inconsistent, and prone to crashing. You'll want to start by consolidating your data. Make sure you have an accurate list of what's in each freezer, rack, and box. You'll want to double check for duplicates or conflicting records, and you'll want to standardize the formats that your data is entered and correct any typos. Once your spreadsheets are cleaned up, you'll want to talk to your software vendor to see if you can import your data directly into the new system. Just a hint, with LabKey, we actually can import those spreadsheets directly and will help clean up that data for you. So you don't have to necessarily do this step on your own. But when it actually comes to cleaning up those spreadsheets, what should it look like, right? There's a lot of different options, and I've seen a lot of different things from labs. But kind of over implementing dozens of clients, I've kind of figured out kind of the best data structured practices to make your life much easier, even if you're staying in spreadsheets or if you're transitioning to a new system. So in this table here, you can see at the top row is reserved for your data field headers. This defines what information you're capturing. The dark blue column is your sample ID. Every single row should have a sample ID. It's your unique anchor point for each sample. The light blue fields represent important metadata fields for your samples. These can include things like patient ID, tube type, or study group. The green columns are for your storage information. And when it comes to storage, you'll notice that I broke these down into different columns for like freezer, shelf, rack, box, row, column. I didn't combine these into one long string because when these fields are separated, it's much easier to sort and filter and analyze your data. If you need to look for all of your samples in a particular freezer or a specific shelf, you can do that in just a couple clicks. It also makes importing data much easier to another system because every system has different requirements. If you need to combine your fields together to import it, it's much easier doing that than separating them if they're already combined. Separating them is time consuming and error prone. But keeping your storage data clean and broken down into clear individual fields, you'll have a smoother transition into any new system, and your spreadsheet will be easier to manage, even if you stay in Excel for now. Um, and one other thing I should kind of point out too is every single sample should be its own unique row. Do not combine samples into one row. It gets messy very fast when you do that, and it's hard to figure out exactly what you have in your system. I won't lie, this step is probably the most time-consuming step. I do this for labs all the time, and I can see definitely how complicated and messy it is. It's also the step when labs actually start to realizing just how bad everything is and how many errors have been made. So this makes them much more eager to migrate than they were before, and they were already eager to migrate in the first place. So let's wrap up with a quick summary of major points we covered today. First, we talked about why good freezer organization is so important and the cost of not having it, whether it's time lost searching, errors in sample handling, or the strain on your equipment. Disorganization has a real impact on your lab's efficiency and accuracy. Next, we looked at common challenges, disconnected records, difficult tracking movement, and poor disposal practices make it incredibly hard to know what you actually have, where it is, and whether it should still be there. We then explored the physical tools you can use to build structure in your freezers, things like racking systems, consistent labeling, and color-coded strategies. These are low-tech, but they go a long way in creating reliable storage systems. We then looked at digital tools and the ways that they can take your system to the next level. Spreadsheets are a start, but purpose-built LIMS and sample tracking software provide visibility, accountability, and automation that growing labs need, especially when compliance or collaboration's on the line. The good news is that every single lab can take these steps to improve, no matter where you're starting from. With the right combination of processes, tools, and commitment, you can transform your freezer chaos into a well-oiled machine. So with that, I want to thank you all for joining me today. I'd like to open the floor to any questions you may have, so leave those in the chat or the Q&A. While you get your questions in, we do have a few services we can help take your control to help you take control of your lab to better organize your freezer. We have Sample Manager, LabKey LIMS, or SDMS, which is a scientific data management system. So if you'd like, we can offer a personalized demo to walk you through how we can help your specific lab. We also offer free 30-day trials. And of course, if you have any questions, always feel free to contact us. We're always happy to help. So I'm going to go ahead and look at questions, but please do let me know if you have anything there. So leave it in the Q&A or the chat. So I'll just pull those up really fast. Let's see. So we're using spreadsheets now. How do we know when it's time to move to a more advanced system like a LIMS? So, a good rule of thumb is that if it's taking you more than just a few minutes to find samples, or if you're managing lots of different samples, it's probably time to start moving. Other signs could be that your spreadsheets are crashing a lot, you're making duplicate entries, you're finding errors, you're having difficulty auditing actions. These are kind of signs that you've outgrown your current setup. What are the most common uh, errors you see when cleaning up those spreadsheets? How worried should we be? So if you're in a spreadsheet, honestly, I would just be worried in the first place. It's so easy to accidentally remove information that you don't mean to. I've done that myself, and I consider myself a more advanced user of Excel. But some of the most common issues that I've seen is that people will put samples in the same location when that's not actually possible. So that's very difficult to kind of find. Um, I also see typos in a lot of these different fields. So if we're looking at a collection date field, I'll see that a sample was collected in 1900. The company was not around in 1900, so that's not possible. I'll also just see data's in the wrong columns, right? You'll see that someone entered in a tube type in the data collection field. So those are the types of issues that we're seeing. Or it's just there's no data there at all. It's all missing. You can't cross-reference anything. Will LabKey help migrating to LabKey and the setup and implementing of Sample Manager? Yep, so that is all included in our services. What we'll actually do is we'll help you kind of look at your historical data. We'll clean it up for you. We'll set up the actual software, how we think best works with you. We work with you at every step of the stage so we can really make sure we're covering all of that support. And then we help train any new users. We either do that in group trainings, one-on-one, we have video references, a whole bunch of things to make sure that you're successful. And then at that point, we're there to support you, answer any questions, help you with new projects that come up down the line. So we really value our partnership and making sure that you're successful with the software. Some vendors charge extra for additional support. What is the cost for LabKey to provide support? We don't charge anything extra for that. That's all included in the subscription. So we really, really value providing that support. We don't think you should get help for using the software that you're buying, right? You should just get that support immediately. Um, does LabKey support international regions? Yep. So we have customers all over the world. We provide support for all of them. So we can really make sure that you're successful. We can have clouds hosted in different regions depending on what your needs are. But we're very familiar with kind of helping those different formats that regulatory standards are needing to be met. Cool. And of course, if you have any other questions, please leave them in the chat. Let's see. So what features does a LIM provide for data entry that a spreadsheet doesn't? A LIMS generally focuses on the data integrity piece and more of that automation pieces. So it helps prevent accidental edits or deletions. You have to purposefully make those deletes or those edits, right? You can't just accidentally and hit the wrong cell and hit a key by mistake and hope your lab tech catches it. It can also provide requirements for the type of data you're entering. So if I'm in a date collection field, I should only be able to enter a date. I shouldn't have free text in that specific space. While with Excel, it can help with providing dropdowns. It's very easy to get around those, right? So we really enforce those kind of restrictions. But then also with the LIMS, it's doing those automated audit trails to make sure that we're capturing all of those changes. Excel, it's very difficult to do that, right? It's you kind of have to restore previous versions. About how long does it take to get organized? So that question can vary depending on where you're at. Are you a brand new lab with not a lot of historical data? Are you a lab with thousands of samples that have piled up over the years? So it really depends on how much time and resources you can also provide in getting organized. In general, when we migrate labs over to our software sample manager, this process takes 6 to 8 weeks. And that's with us going through your historical data and cleaning it up, right? We've practiced this. We have the expertise on the best methods of doing that. Um, we also train the new users on how to use that software and kind of make sure we know what the best practices are for managing that data. As you can imagine, if you were doing that on your own, that process might take a lot longer. It also depends on exactly what you're wanting to implement. Are you wanting to go through and audit every single sample in your system? That takes a lot of time, but having the right tools and the right connections and resources and expertise can help reduce the amount of time that it takes there. What's the best way to get staff to actually follow new organization processes? You want to make sure that the new system is easy or easier than the one that's currently in place. You want to build it into existing workflows and provide hands-on training. Most importantly, you want to show them why. Like, is it going to be faster to search for samples? Will there be fewer errors? Is it going to be less stressful? And you don't want to forget about involving them in the planning stage, they're more likely to adopt a system that they've helped shape. Um, let's see. How do I get leadership buy-in to invest in sample tracking software? So, really, you want to frame it as a cost avoidance or risk reduction strategy. You want to emphasize the time that's saved, the reduction in compliance risk, and improved data integrity. If possible, you want to show them a concrete example of a mistake or inefficiency in the lab you've encountered and how it could have been avoided, right? Most leadership teams will respond well to stories that are backed up by numbers. So you kind of want to prepare that and work with them and kind of show how it's a priority. But if there's any other questions, also feel free to stay in the chat. But thank you so much for joining us. Really appreciate it. Always reach out. My email is lancel@labkey.com, so feel free to email me if anything comes up afterwards. Otherwise, we will be emailing a recording of this webinar out, as well as some more information on how to really get organized with your freezers and how we can help out. So thank you again, and have a great rest of your day.